For the Starch Agar test, the materials that you will need to gather for it include an inoculating loop, Bacto incinerator, your unknown bacteria, which is your slant stock culture, your starch agar plate, which is right here, your iodine solution, and a plastic pipette dropper. To start the experiment, you will sterilize your inoculating loop by putting it in the Bacto incinerator for about 5 to 10 seconds. Once you've done that, allow the loop to cool off. Once the loop is cooled off, grab your slant stock culture of your unknown bacteria, take the cap off, flame the tube for 5 seconds, and then stick your sterilized inoculating loop in, scrape out some of the culture, flame the test tube again for 5 seconds, put the cap back on, put it back on the test tube rack. Now what you're going to do is you're going to use the streak method to spread your stock culture onto your starch agar plate. So just do it like so. Going to spread it around the whole plate. As you can see, I'm going from left to right. And each time that I spread, I actually start back from where I last had spread it. Now I'm going to close it, sterilize my inoculating loop. Once I've sterilized it, I can put it back down on my bench top. Alright, and the next step that you're going to do is you're going to grab your iodine solution, which is this, and then you would use your plastic pipette dropper to obtain some of the solution out of it. So we're just going to pretend like I got some of it out of there. And of course, if this was a real lab, your pipette dropper would actually be sterile. But since this isn't a real lab, I'm just using the plastic pipette dropper that I have on my benchtop. So once you get that, you're going to open the lid of your starch agar plate and you're going to flood it with this iodine solution. And once you've done that, close the lid. I'm just going to go like this to make sure I've spread it all along it. And then once I've done that, let the culture incubate for about 5 minutes before you analyze anything. And of course, in a real lab, we would get rid of this in the biohazardous container. After you have analyzed and observed your culture and got the results that you needed, you're going to get rid of it. You're going to put it in the biohazardous waste container, just gone. And then you're going to decontaminate your workspace. You're going to get rid of all these materials, get them out of the way, spray it down with germicide, wipe it down with paper towels, make sure you throw the paper towels that you used away in the biohazardous waste container. Then you're going to remove your gloves because they've been contaminated. You're going to put those in the biohazardous waste container as well, and then you can wash your hands. And then grab a new pair of gloves because we aren't done yet. We're moving on to the blood agar.